Bree, can you hear me?
Yep.
Good. Okay, Bree, can you see that all right?
Yeah. I downloaded the Socrative app, whatever it's called. And it says, like, as soon as I open the app, it says student login. So will you give us the room name to take the quiz?
Oh, yeah. Yeah.
Okay.
Okay, Bree, do the slides move?
Yeah, they're good.
They're good? Okay. Goodness, when that thing, oh, no, it's okay. Come on, blue dot. Go away, go away, go away. So we need to go. Oh, there you go. It takes one of these. Okay, Bree, I will give you, I'll give Mrs. Schaefer a copy of the quiz. When you get back, you can take it from her.
Okay.
I think that's everything. All right. We have people who have not taken the quiz yet, so I can't go over it. If I mute it, the recording will still pick it up. So, and if I turn off the recording, getting the recording back on. So I turn it off, Zoom starts to save it. So I will go over the quiz when I give it back on Monday. All right, you guys are writing good questions. Keep writing those questions. All right, back to metabolism. So let's review. You take monosaccharide sugars like glucose, but other monosaccharides can feed into glucose. You can also feed fructose or galactose or mannose into glycolysis as well. So you put into glycolysis monosaccharide sugars, typically 6 carbon sugars. What do you get out? What do you get out? Rebecca, what do you get out? Yes.
Sorry.
You're Rebecca, right? Yeah. You put monosaccharides into glycolysis, what comes out? Just name one thing that comes out. Brooke? 2 ATP, right? And that's net. You actually have to burn 2 ATP to prime the pump. You actually make 4, and 4 minus 2 is 2. So you make 2 net ATP. All right? Since I'm picking on Rebecca's, I'll pick on this Rebecca too. Name me another thing that comes out of glycolysis. That's Krebs cycle. Okay, glycolysis. Glycolysis. You were right for the Krebs cycle, but glycolysis. Right, 2 ATP, and then there's 2, what else? NADH, 2 reducing equivalents in the form of NADH. Very good. And then, Kristen? 2, Caitlin, all right. That's close.
Carbon dioxide.
No, that's Krebs cycle. That's Krebs cycle. No, that's Krebs cycle. The 3 carbon dudes at the end, what are they called? You have your hand out, Lydia? It's on your hand out. Bree's got her, she's staring at her hand up there. Do you know, Bree?
Is it
Pyruvates. Pyruvate. Pyruvic acid, pyruvate, 3 carbon compounds. So you get 2 3 carbon compounds, pyruvate, and 3 plus 3 is 6, so that's where your 6 carbons went. When you do metabolism, try to count carbons. If you're missing carbons, you left something out. So 3 and 3, that's 6. Now in eukaryotes, where do those pyruvates go? Where do they go? Do they stay in the cytosol? Do they go to the nucleus? Do they get excreted? Where do they go? Which organelle makes the energy in eukaryotes? Mitochondria. Since these reactions are catabolic, they occur in the mitochondria. Glycolysis occurs in the cytosol. All subsequent reactions occur in the mitochondria. Bacteria don't have mitochondria, so all this happens in the cytosol. So the next stage is you take pyruvate, you convert it to acetyl coenzyme A. That gives you 1 carbon dioxide, which you expect, because acetate has 2 carbons, pyruvate has 3, that's where the other carbon went, 1 carbon dioxide. And then you also get and NADH out of that. So that means for every 2 sugars, every single glucose, excuse me, you'll get 2 pyruvates and 2 acetyl coenzyme A's out of that. Now, what's the enzyme that catalyzes this reaction? It's that big thing, big complex in mitochondria and also in bacteria. Its abbreviations are PDH. Sydney, got it written down there? Sorry? Peptidoglycan is what's the cell wall of bacteria are made of. Right, you know. Got your letters a little mixed there. Cassidy, any ideas? Jess? Hey, Bree. Sorry? No. Maddie's got her hand up. I think she knows. Pyruvate dehydrogenase complex. It's a complex. There's several different polypeptides. Pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex is dysfunctional in some people. It causes a very severe genetic disease. When it does, well, I get the privilege of teaching you guys genetics next semester, so we'll go over quite a few genetic diseases. And if any of you end up doing peds or family medicine, or maybe even oncology, you'll do quite a bit of genetics in your practice. So learning that type of genetics is important. So pyruvate dehydrogenase complex converts pyruvate into acetyl coenzyme A. What's a vitamin that's used in the making of acetyl coenzyme A? It's fortified in milk. Vitamin D, yeah, there's vitamin D in milk, yes, but it's not used to make coenzyme A. What's the other one? Did someone say it? Janet, did you say it? No? Pantothenic acid. Pantothenic acid, otherwise known as pantothene. Pantothene. And if any of you went and read milk cartons since last week and saw that, well, kudos to you for chasing after that. All right, now we get to the Krebs cycle. So the Krebs cycle is not a linear pathway like glycolysis, it's cyclical. So your first substrate is a molecule called oxaloacetate. Oxaloacetate condenses with acetyl coenzyme A, and you make citric acid. Citric acid then goes through a series of oxidations and rearrangements. And in this cycle, you end up regenerating oxaloacetate. In the process, you produce 2 molecules of carbon dioxide. You produce 3 molecules of NADH per acetyl coenzyme A. You produce 1 reduced flavine, and you produce 1 GTP, but energetically that's equal to an ATP. So if I ask you how many ATP does the Krebs, 1 turn of the Krebs cycle produce, it's just 1. You might say it doesn't produce any ATP, it produces a GTP. Well, in your head, GTP equals ATP. So we'll just say that's an ATP. And this is shown right here. So 3 NADHs, 1 FADH2, and 2 CO2s, count them 1, 2, and an ATP. All right, let's rehearse. Jenna, what goes in to the Krebs cycle? Say that again?
I don't know what
Acetyl coenzyme A. Acetyl coenzyme A. So it's acetate. So vinegar. Do you use vinegar on your salad dressing? I don't eat salad. Not vinegar. Okay. I'm an oil and vinegar guy myself. So vinegar is acetic acid. Acetic acid has 2 carbons. This molecule is virtually the same structurally as acetic acid, so it's vinegar, as the major component in vinegar. Vinegar has other spices in it and things like that. So, but the main component is acetic acid. So if you want to think of this as vinegar, I won't fault you. Just don't say vinegar on a test, okay? That would be half credit. So it's acetyl. That's like the preposition form or something of acetate. Coenzyme A, which we're just going to write like that. And coenzyme A is a carrier. It's a carrier. When you change a socket in your house, do you unscrew the screws barehandedly? No, you use a screwdriver. This is kind of like the screwdriver that holds the screw. Okay, so that'd be the screwdriver, that would be the screw. This is the thing that's going to, this is just a carrier. Because when you hand this off to citric acid, the coenzyme A gets released and gets recycled. So all that does, it's a carrier, that's all it is. All right, so you put in, say it, Janet, what's it called? Acetyl coenzyme A, and then it condenses with oxaloacetate. Say it. Oxaloacetate. So this molecule here, that's oxalate. Patient comes in to you, oh, miss, ow, my stomach, I'm puking, I got, ow. I got blood in my pee and kidney stone. Most of your kidney stones, the vast majority of kidney stones are oxalate. Oxalate's a breakdown product of vitamin C. So people who take horse pills of vitamin C are at higher risk for kidney stones, unless you're drinking lots of water like you're supposed to. The other place where you see oxalate is in things like spinach leaves and things like that. So if you're drinking that, eating that stuff, which you should, spinach leaves are good for you, should be drinking lots of water. So oxalate, if you take oxalate and you add to oxalate acetate, so essentially, there's your acetate, here's your oxalate, so it's oxaloacetate. The chemistry, I'm just showing you why it's called what it's called. It's not called that to torture students. There's an actual reason why it's called that. So say it again, oxalo. Good. And then you make, Sydney, citric acid. Therefore, this cycle is also called the citric acid cycle. Say it. Good girl. Citric acid cycle. You run it through and you regenerate the oxaloacetate. And then, Lynn, I got it right. Your products are, maybe just one of your products. No, 1 ATP. 1 ATP per.
Oh, I was sorry. 1 ATP.
Okay, and then what else? Yeah.
1 FADH.
1 FADH, reduced flaving, and then where your carbons go? 2 CO2s. That's what comes out. That's what comes out. You have burned the carbon all the way to the most oxidized form of carbon, carbon dioxide. You started with glucose, 6 carbons. Remember, you're going to put 2 of these per glucose into the cycle, into Krebs cycle, and that will make 4 of these. You generate 2 CO2s per 2 of these. That's 2 for all 6 of those carbons are now completely oxidized to CO2. Any question?
I was just confused about, because you said to like keep track of the carbons, and I'm just confused about all the carbons, because there's like a bunch of them.
Right. So there's 2 here, right? This turns twice per glucose, right?
Yes.
So that's 4. And then for every PDH catalyzed reaction, you release 1 CO2, but you have to do 2 of those because 2 pyruvates come from glycolysis. So that's 2 CO2. So 1, 2, 3, 4, 5, 6. You see where that comes from?
What about the other ones, the century and the, see all those?
These are just part of the cycle that regenerate the oxaloacetate. This is a cyclical cycle. It's a cycle. You form your first product citrate. The rest of the cycle is to regenerate the oxaloacetate so you can do it again. You pass go, collect them colors, and do it again. Yeah?
You said that it turns twice to get glucose.
To oxidize glucose completely, it has to turn the cycle twice because you get 2 pyruvates from glycolysis, don't you? So that'll generate 2 acetyl coenzyme A, right? So you have to feed both of those into the cycle. That's why to completely oxidize the glucose, you have to turn the cycle twice. That's a Krebs cycle. Yes.
So then in reality, like after, for 1 glucose, basically you get 6 NADH, 2 FADH. Okay.
Yep. And in fact, that's a nice little intellectual exercise to do. If we were to take the products of glycolysis plus Krebs cycle, so per glucose then, if we take a look at glycolysis, we have 2 NADHs. Then we look at the pyruvate dehydrogenase catalyzed reaction. We're going to get 1 for each reaction, but for every pyruvate that's fed in, we're going to have to run this reaction twice. So that's going to be 2 times 1. So that ends up being 2. And then for the Krebs cycle, you get 3 but you have to run it twice, and then 1 and then you'll have an ATP. We'll just say it's an ATP, and so on and so forth. So if we add all that up, we get 6 plus 2, that's 8 plus 2, that's 10. We have 10 total NADHs, right? We have 2 of these. We have 6 carbon dioxides, which that makes sense since 6 carbons per glucose went into the cycle. In terms of ATP, we end up having 2 plus 2, that's 4. And I think that's everything. Did I leave anything out? Now, if that's what this cycle does, you have a problem. And the numbers are nicely over here, and it shows you essentially what's what and so forth. These electron carriers are reduced. If you're going to continue to run the cycle, you need to oxidize those. The rest of metabolism is all about what to do with these high energy electrons. Now, in aerobic organisms, you put them into this thing called a electron transport chain. The electron transport chain sends the electrons down the chain to molecular oxygen. Molecular oxygen is reduced to water. That will reoxidize these carriers so that you can go on to metabolize more glucose. But what happens essentially to these electrons is the following. So electron transport chains are always embedded in membranes. Always. What's the main membrane in a bacterial cell? What's the main membrane in a bacterial cell? Sorry? The cell membrane. So this electron transport chain is embedded in the cell membrane. In eukaryotes, this is embedded in the inner mitochondrial membrane. Because mitochondria, remember, have two membranes, an outer and an inner membrane, and it's embedded in the inner mitochondrial membrane. But in bacteria, it's embedded in the cell membrane. So the cell wall is out here. For an electron transport chain to work, you need three main things. First thing you need is a cell membrane, because the components of the electron transport system are embedded in the cell membrane. Second thing you need to alternate your electron transport chain between entities that carry both protons and electrons and entities that just carry electrons. You need to alternate between entities that carry both protons and electrons and carriers that just carry electrons. So you need a cell membrane because the components of the electron transport chain are embedded in the membrane. You need to alternate those carriers between things that carry protons and electrons and things that just carry electrons. And you need to topologically arrange those carriers so that when a carrier that carries both protons and electrons gives its electrons to the next entity, the protons are extruded onto the other side of the membrane. So you need to have a cell membrane. The components need to be embedded in the cell membrane. You need to alternate between carriers that carry protons and electrons and carriers that just carry electrons. And you need to topologically arrange those entities so that when protons and electrons are passed to the electron-only carriers, the hydrogen ions are extruded to the other side of the membrane. You need those three things to have an electron transport chain. And you have those three things in the electron transport chains of organisms. Now, the flow of electrons down the electron transport chain is also thermodynamically spontaneous. In chemistry, you might have, remember this quantity called delta E? Remember that from chemistry? Does that ring a bell? Does that summon up visions of mathematical terror? So delta E is a measure of how likely a molecule or ion is to accept or give up electrons. If delta E is very positive, it tends to accept electrons very vigorously and not give them up. If delta E is very negative, the entity tends to give up electrons easily and not hold them very, very tightly. So at the beginning of the electron transport chain, your carriers have negative delta E's. As you go down the chain, the delta E of each carrier gets more and more positive until down here, you have molecular oxygen. That's your acceptor. The delta E of a molecular oxygen is very positive. Therefore, the transfer of electrons down the chain is thermodynamically spontaneous. Thermodynamically spontaneous. Okay, let's slam the truck in reverse. Let's back up a little bit. I'll start in back this time. Rookie, morning. So name me one thing you need for electron transport chain. Cell membrane, and the components of the chain embedded in the cell membrane. Very good. Rebecca, what's another thing you need for electron transport chain? A little hard, these old ears can't hear. You'll need hydrogen ions, yeah. You'll need hydrogen ions, but the chain, what does the chain need? The electron transport chain. Proteins that are carriers. Not all the carriers are proteins. Coenzyme Q is a lipid molecule, but it interacts with proteins. So proteins are some of them. Okay, what about those carriers? What's unusual about those carriers that you need to have for electron transport chain? Yeah, they alternate between those that carry just electrons and those that carry both protons and electrons. Macy, what else does that chain need? You can use your notes.
Do you, at this point, you are proteins?
Yeah, many of the carriers are proteins. So we have membrane, they're embedded in the membrane. They alternate between those that carry just electrons, those that carry both protons and electrons. And the third thing is, how about their arrangement in 3D space? Brooks one has got the best notes back there.
Hydrogen ions.
They have to be arranged so that hydrogen ions are extruded.
Yeah.
Extruded, say it. Extruded, released, pumped from one side of the membrane to the other. Right? That's very important. Bree, what can you tell me about the thermodynamics of transfer of electrons down the electron transport chain? No, it doesn't follow a concentration gradient. Not a concentration gradient. It goes from carriers that give up electrons very easily, gradually down to carriers that don't give up electrons at all. So the transfer is thermodynamically, what's the word we used? Oh, you're such a spontaneous person. You're thermodynamically, thank you, spontaneous. Thermodynamically spontaneous. Yeah, it's not a simple thing to do early in the morning. Let's look at some of the carriers. So the carriers include flavoproteins. So flavo It's derived, their cofactors are derived from riboflavin, hence the need for riboflavin in our diet. Bacteria, many bacteria make their own riboflavin. You don't need to get it from the diet, but some need to get it from their diet. NAD dehydrogenase, that's the first thing that NAD gives its electrons to. Coenzyme Q10, also known as ubiquinone. If you go into health food stores, for example, if you go into Jackson Crossing and you go down towards the Target, on your right, there's a health food store. And if you walk in there on the right-hand side as you walk in the store, at the middle of the shelf, you'll see coenzyme Q10. Coenzyme Q10 is this. Some athletes take it before races. Any athletes there? Have any of you heard of supplementing with Q10? It used to be all the rage. It's not so much anymore, but it used to be a big deal. Any of you heard of it or taken it or what have you? No, Natalie, no. It used to be a big deal. It's not so much anymore. Back in the 90s, everyone was supplementing with coenzyme Q10. And was getting a serious case of the trots. So the rationale for taking that would be that you improve the efficiency of your electron transport chain. That's the rationale with taking it. Now this is a lipid soluble molecule. Because it's lipid soluble, it moves very easily within membranes. So this acts as an electron shuttle between carriers. And then another group of carriers that carry just electrons are cytochromes. These are very important. These are iron-containing molecules, hence the big need for iron in your body. They have that iron, they accept electrons, and then they give those electrons up very, very easily. So cytochromes, flavoproteins, coenzyme Q10, and NAD dehydrogenase. Those are the main players in the electron transport chain. So let's look at how this works. So there's your membrane. Your first carrier is an L-shaped molecule. It's a complex of many proteins. It is known collectively as complex 1. Complex 1. Complex 1 is going to accept it accepts protons and electrons from NADH, and it gives those electrons to coenzyme Q10. In doing so, it will pass the electrons to a series of carriers. The first ones accept protons and electrons. The second ones just accept electrons. So what happens when it passes those electrons? These hydrogen ions get extruded out here. Coenzyme Q10 binds electrons with protons. So now it has to pick up protons from the cytosol. Already, we have protons essentially being moved from one side of the membrane to the other. Biological membranes, remember, are impermeable to hydrogen ions. So this is a way you pump those hydrogen ions across the membrane. Q10 is now going to give its electrons and protons to something called complex 3. Complex 3. Complex 3 will take the electrons and the protons and get them to a series of cytochromes. Those series of cytochromes, remember, just take electrons. They don't take protons. So this is going to extrude more of those protons. And complex 3 is going to give these electrons to a small protein called cytochrome C. Cytochrome C. Cytochrome C is going to deliver those electrons to another complex in the membrane called complex 4. Complex 4 is also known as cytochrome oxidase. So this will deliver electrons here. This will shuttle back and forth like that. This will take the electrons and it will take molecular oxygen and convert it to water. So you need to take 2 more, actually you'll need 4 of these in there, and you'll make water. And then you'll make 2 of these. You've pumped these protons out here. Those poor lonely protons, they want to get back home. How are they going to get back home? You've essentially created a charge and concentration gradient on either side of the membrane. That charge and concentration gradient can be used to do work. You've essentially made a mini battery, haven't you? All by throwing electrons through this chain. You've pumped all these protons out into the space. And that concentration and charge gradient is called proton motive force. Proton motive force. Proton motive force consists of the charge and concentration disparity on either side of the membrane. In order to let these protons back into the cell will exact a price. So there is also embedded in the membrane a very large, massive protein complex That's ugly, but I think you get the point. It's called the ATP synthase. It consists of a pore and these globular bits, there's 3 of them. And these protons are going to come through here. As they come through here, this will take ADP, an inorganic phosphate, and convert it to ATP. So you'll let the protons back into the cell. As you let the protons back into the cell through the ATP synthase, you make ATP. That's the price that the cell will exact for bringing those protons back into the cell. This will make bucket loads of ATP. That's why aerobic organisms will grow so much faster than anaerobic organisms, because you are essentially using these high energy electrons to create a proton gradient across the membrane. The proton gradient is used to make ATP and to make lots of it. Lots of it. So you make about 2.5 ATP per NADH. You make about 1.5 ATP per FADH. So go ahead and calculate how many ATP you're going to make per glucose. Go ahead and do that right now. Come up with a number. You make 10 NADH and 2 FADH2s per glucose. You make 2.5 ATP per NADH and about 1.5 for every FADH2. Go ahead and calculate how many ATP you make.
How many FADH?
2.
Wait, is it 2 or 2.5? ATP?
2.5 ATP per NADH. That's based on the best stoichiometric calculations available to us today. Those numbers are not final, but they're the best we have for now. Did you get a number? How many? $28? That's close. Anyone get a number other than 28? 34. So 10 times 2.5 is 25. 1.5 times 2 is 3. So that's 25 plus 3, that's 28. Then there's the 4 that you made in glycolysis and Krebs cycles, that's 28 plus 4 is 32. And where'd you get the other 2? No, 4. 4. So it's about 32. So it's in the range 30 to 32, something like that. Your old biology textbook said it was probably 38 or 36. That's the number you see in high school books. That's wrong. That number is wrong. That number has been known to be wrong for the last 2 decades. Why they keep putting that number in biology textbooks, I don't know. But they do, and it's wrong. So this mode of making ATP through electron transport chains that make proton gradients that are then used to run the ATP synthase. This is called oxidative phosphorylation. Otherwise known as cellular respiration. Cellular Respiration. And herein lies the reason why we need to keep breathing, because oxygen is our terminal electron acceptor. Without oxygen, we don't make ATP. In order for life to continue, cells need a constant source of energy. Without oxygen, you don't get ATP. Without oxygen, cells can't make the energy they need to survive. Without oxygen and without the energy to survive, cells start to die. That's why we have an absolute requirement for oxygen. The only cells in your body that don't have an absolute requirement for oxygen are cells that don't have mitochondria, like red blood cells. Red blood cells depend very heavily on glycolysis. All right, let me pause right here and see if there's any questions. We'll go ahead and take our break right now. We'll resume at 20 till. And if any questions come up, we can ask those then, and then we will move on to those that don't use oxygen. You brought it. Good.
Should I just leave it right here?
Yeah, I know. No, just-
Okay. Thank you.
So Kenny, you can drop it at my office if that works. Yeah, it's more than the culture health is when it's young. It's that culture is growing. When it's growing, cells are foraging for foods and life. They get to stationary phase, and they're trying to conserve energy. Other choices face complex diseases. It's people with AIDS. When you face complex diseases, those are acid mycobacterial tuberculosis. You'll probably in practice do it. So, nice complex. That's avium, mycobacterium, avium, intracellular. It used to be called scroffic disease. Avium golvis and intracellular.
Thank you so much. Great. Except keep the door open, not be close to the temperature. It's still bloody cold.
It's always pepper spray. Yeah. Oh, okay, it's not pepper spray.
No.
Oh, yeah. So I would say. Whatever.
Did it stand?
You always wear socks.
Very good. It is time to resume. Now, this picture from your book illustrates what I've been trying to explain, that you have the carriers embedded in the membrane, they exchange electrons and send them eventually to molecular oxygen to produce water. In this transfer of electrons down the electron transfer chain, protons are pumped from the cytosol into the periplasmic space. Those protons will then re-enter the cell through the ATP synthase, and the re-entry of protons will drive the synthesis of ATP from ADP and inorganic phosphate by the ATP synthase. Some organisms that are anaerobes still do this process, but instead of using oxygen as a terminal electron acceptor, they use nitrate. Nitrate is then reduced to either nitrite or to atmospheric nitrogen, N2. We will do experiments in the laboratory for nitrate reduction. Many organisms that live in soil can use nitrate as a terminal electron acceptor when the soil becomes waterlogged. The conversion of nitrate to atmospheric nitrogen is a process ecologically called denitrification. Denitrification. And denitrification is responsible for most of the nitrogen loss in soil. Another group of organisms can use sulfate as their electron acceptor. They will reduce sulfate to sulfide. Sulfate will be reduced to sulfide. Hydrogen sulfide smells like rotten eggs. And if you've been around organisms like this that do this, the laboratory has a particular aroma that you just have in that experience. The conversion of sulfate to sulfide, so-called sulfate reduction, is exquisitely oxygen sensitive. Organisms that do that live in very oxygen poor environments, and they will usually die even if they get a whiff of oxygen. Organisms that do that, usually their names begin with desulfuro, desulfuromonas, desulfurobacter, desulfurospirilla, desulfuro, your mother-in-law, whatever. These, you see desulfuro, it means it's a sulfate reducer. Those are very important in marine sediments and other types of sedimentary environments where you have very low oxygen tension and high sulfate concentration. Rice plants, for example, rice plants will produce quite a bit of sulfate. And so the sediments in the soil will be converted into sulfide, which is then used by the rice plant for its sulfate. It's sulfur uses. All right, so this type of situation, that is a form of anaerobic cellular respiration. Most organisms that do cellular respiration use oxygen as their terminal electron acceptor and are therefore obligate aerobes. Obligate aerobes. So whereas this occurs in the cell membrane of bacteria, in eukaryotes, this occurs in the inner mitochondrial membrane, and then protons are pumped into the intermembrane space and then pumped back into the mitochondrial matrix. Mitochondrial matrix. And we talked about that process. All right. So aerobic respiration has a tendency to throw reactive oxygen species like hydrogen peroxide and like superoxide radicals. Remember your cat in your room, squirrels in your room? Remember those? And so cells have these molecules that deal with the cat in your room and the squirrels in your room. Superoxide dismutase, which is abbreviated SOD, and catalase. They work in a 1-2 punch. The superoxide dismutase converts superoxide radicals into hydrogen peroxide, and then catalase converts hydrogen peroxide into molecular oxygen and water. Some microorganisms lack these enzymes, so they don't do quite as well in oxygen. Streptococci are among those organisms that lack catalase. Now we will do a test in the laboratory for catalase because catalase produces molecular oxygen. To see if the organism has catalase, you put a drop of hydrogen peroxide on the cells. And if catalase is present in the cells, the culture will start to bubble vigorously. It'll bubble and foam and what have you. If it doesn't have catalase, you may get the occasional bubble here and there, but you don't get the vigorous bubbling. That's a very easy test to do because streptococci are typically catalase negative. And so that is a test that is easy to do and can distinguish between certain types of bacteria. Other types of bacteria that are catalase negative include members of the genus Lactobacillus. The lactobacilli are important in the food industry because they're important in food spoilage and also in making certain things as well, making certain cheeses and dairy products, kefirs, other types of cheeses, things like that. Bless you. Nitrate reduction requires the presence of an enzyme called nitrate reductase. So instead of cytochrome oxidase, organisms that reduce nitrate, it'll hand its electrons from complex 3 to nitrate reductase. Organisms that can perform nitrate reduction, they're genes that encode the nitrate reductase complex. They are only expressed in the absence of oxygen. There's a very sophisticated mechanism inside cells that keeps those genes off in the air and turns them on when there's no air. Because when there's no air, it's a pretty good shot if you're living in soil or sewage, that nitrate is about. And that nitrate will be reduced to nitrite and then possibly to nitrogen. Nitrate reductase. And that leads to the production of nitrogen gas from nitrate, and that is this process called denitrification. Denitrification. Your book makes the point, and it's a point well worth making. Organisms that are nitrate producers will not grow as fast as they do in the air. Because nitrate has a delta E that's less positive than molecular oxygen. So you don't transfer as many electrons to it, you don't pump as many protons to it, therefore you don't make as much ATP. So when you reduce nitrate, you don't grow as fast as you do if you're reducing oxygen. But you still grow faster than if you weren't doing oxidative phosphorylation at all. Now, what about organisms that don't do any of this business here? In that case, you're going to make all of your ATP through the reactions of glycolysis and reactions like it. Such reactions that yield ATP that don't involve membranes and proton gradients, that's called substrate level phosphorylation. Substrate level phosphorylation. In the case of substrate level phosphorylation, you make some sort of high energy phosphorylated intermediate. That high energy phosphate bond is then transferred to ADP to make ATP. That's how you make ATP during glycolysis. And that method, that mode of making ATP is called substrate level phosphorylation. If, as an organism, you make all your ATP from substrate level phosphorylation, you carry out a fermentation. That's why I want you to divorce your thinking from, if you ferment, then you make ethanol. No. You might make ethanol, but you might make lots of other things too. Fermenters tend to not grow very fast. They don't make very much ATP for glucose. They're used by organisms that do not have an electron transport chain. Some organisms will ferment if there's no oxygen. You give them oxygen, they can revert back to oxidative phosphorylation. But they're going to take those reduced electron carriers. They're going to dump the high energy electrons on the carbon skeleton they generate. They will therefore release reduced organic acids and reduced organic alcohols. They'll produce reduced organic acids and reduced organic alcohols. All right, so if you look at page 6 of your handout, and this just restates essentially what I said before. The NADs are recycled to enter glycolysis. You dump them on the pyruvate, you dump them on the whatever, and you make things like lactic acid or ethanol or propionic acid or butanol or what have you. These are some examples of fermentations. So one of the fermentations that cells will carry out, and you see it here on page 6, you make pyruvate, you turn right around and you give those electrons back to pyruvate. If you reduce pyruvate, you make lactate or lactic acid. This is precisely what your muscles do under anaerobic conditions. So let's say Natalie is on the basketball court and she has to run because coach says, we need to run it, we're gonna run. So you guys, your offense that you're running back and forth across the court. And it's at that time you get very thankful that coach killed you guys during practice. The wind sprints up and down the court. And pretty soon, the other team calls a timeout because you're whipping them with this strategy. You get into the timeout, you squat down to listen to coach. Ow, your muscles are starting to cramp. Why are they starting to cramp? Because under anaerobic conditions, and you're making lots of lactate. Because you can't deliver oxygen fast enough to the muscle, so the muscle starts making lactic acid. And excessive lactic acid built up in the muscle will cause cramping. That is called a lactic acid fermentation. Organisms that do this, you've probably experienced them. Have you ever picked up the milk and, ooh, yikes, you taste it, oh, that's off. That's what the British would say. Or you'd say that's sour, that's spoiled, that rancid taste of milk that's been spoiled. And if you had that unpleasant experience where the milk was at the back of the fridge and you didn't pay attention to the expiration date, then they never, oh, okay, yes, it had. That's lactic acid in the milk. These are organisms that undergo a so-called lactic acid fermentation. That's what causes the rancid taste in spoiled milk. Other organisms will take pyruvate, they'll convert it to acetyl coenzyme A and formate, and then they'll take the acetyl coenzyme A and they'll use it to make an ATP and they'll make acetate. That is an acetic acid fermentation, an acetic acid fermentation. Acetic acid fermentations are found in organisms that spoil wine. You ever heard of this expression? If you don't keep the wine properly, it gets converted to vinegar. That's due to groups of bacteria called acetobacter. Acetobacters can grow in high levels of ethanol and any of the extra sugar in the wine. So dessert wines will have more sugar in them. They take the sugar and they ferment it to acetate. And they convert the wine to vinegar. That's an acetic acid fermentation. And then others, of course, can take acetyl coenzyme A, convert it to acetaldehyde, and then to ethanol. So you have two enzymes there, alcohol dehydrogenase and aldehyde dehydrogenase. And that is your ethyl alcohol fermentation. That's the one conducted by yeast and other organisms too. And then finally, you can see this other arrow here, phosphoenol pyruvate, or PEP. That is the penultimate step before pyruvate. That gets shunted over here, and you make this organic acid called succinate. Succinate. That is a succinyl acid fermentation. So look at the potential end products of these fermentations. 1, 2, 3, 4, 5. Lactate, ethanol, acetate, formate, succinate. So fermenting organisms make ethanol. Oh, really? Yeah, they make some ethanol, but they make lots of other things too. So please don't get the idea that fermentation means ethanol. Fermentation means a lot more than ethanol. They can make lactic acid, they can make acetic acid, they can make formic acid. You get an ant bite, an ant sting, it gets really necrotic, that's due to formic acid in the sting. They can make succinic acid and they can make acetic acid as well. All right, take a look at page 7. Tuesday and Wednesday, we're going to do some tests called the MRVP tests, the methyl red Vogts-Proskauer tests. This is part of the MVIC tests. If any of you do public health nursing and you have to do water sampling, MVIC tests will be incredibly important because MVIC tests will tell you if your water's been contaminated by sewage. So MVIC tests, one of the tests is the MRVP test. MR stands for methyl red test. Methyl red detects the mixed acid fermentation. Mixed acid fermentation. So if you look at the mixed acid fermentation, you have, go ahead and put a check mark by all of these. You have succinate, that's an organic acid. Lactate, that's an organic acid. Formate, that's an organic acid. And acetate, that's an organic acid. 1, 2, 3, 4 organic acids. What's that going to do to the pH of the medium? It's going to lower it. So if you put the acid base indicator, methyl red, into this medium, it's going to turn red. That is the methyl red test. You're looking for the mixed acid fermentation because it makes a variety of mixed acids. And E. coli and other organisms like E. coli perform the mixed acid fermentation pathway. The other pathway is the butanediol fermentation pathway. The butanediol fermentation pathway. Let's look at this one. With this one, you make ethanol and 2,3-butanediol. Those products are pH neutral. So if you put methyl red in this test tube, it will not turn red, it will stay clear. Instead, you will do the Vogts-Proskauer test, the VP, think Vice President. You'll do the Kamala Harris test. And you will look for 2,3-butanediol. You will add a reagent called alpha-naphthol. Alpha-naphthol combines with 2,3-butanediol and turns the test tube a beautiful Alicante wine red color. Have you seen Alicante red wine? It's this gorgeous burgundy red color. It's just, I grew up on a grape farm. Was exposed to all kinds of wines and things like that. My parents were big wine people. It comes with growing up on a grape farm. So it's part of Alicante wine is it's from the Alicante area of Spain. And it's very famous because the climate there is very good for growing wine grapes. Anyway, you get this beautiful wine color with that. That's a positive. If you mix acid fermentation, it'll be clear with the VP test. But with this, you get that beautiful Alicante wine color after about 10 minutes. That's a Vogts-Proskauer test. This is part of the MRVP test that we will do Wednesday and Friday. That distinguishes two different types of fermentation. Two different types of fermentation. Now on page 9. This is an unusual fermentation here. Notice that the product of this fermentation is propionic acid, propionate. So to circle propionate, this is called the propionic acid fermentation. Why is this important? Because there's an organism, it used to be called Propionibacteria. Its name was changed. It's now called Cutibacterium. Cutibacteria acnes. Acnes causes acne. And so when pimples burst, you take your oxy scrub and you scrub, and it bursts. And you get that white stuff that comes out of the pimple, or if you hit it, or if you squeeze it, which you're not supposed to do, but everybody does. And you get that stuff and it's got that musty smell to it. That musty smell is propionic acid. That's made by Cutibacterium and its compatriots. That's the fermentation essentially that's causing this. And acne and other types of acne, some of you again will work with peds and family medicine. You'll have adolescents with acne problems will come in. That's the bacterium that's causing their woes. That produces the commodes and things like that. All right, there's just one more fermentation I want to call your attention to. Oh, yeah, here we go. Page 5. This fermentation here is what Clostridia do. Clostridia all ferment. But Clostridia will produce butanol or they'll produce butyric acid. So let's say some of you have had this experience. You take your gym clothes go to the bottom of your gym bag. Your clothes are all stretched up, or you put them in a plastic bag, and then you leave them over the weekend, you forgot about them. You open them up on Monday, and the smell just hits you right in the face, practically knocks you off your feet. That dirty sweat sock smell, that's butyric acid. Those are Clostridia. They're thriving in the anaerobic environment of your gym bag or your sealed bag. They're fermenting amino acids and sugars from your sweat and producing butyric acid. So Clostridia cause several types of diseases. And this is the fermentation they perform. They make butyric acid. It's a four carbon organic acid, and it's gestunken. It smells awful. So those are fermentations. And fermentations are used industrially to make things that we want. All right, now we've talked about glucose. Let's talk about other molecules. Lipids. Go to the back page of your handout. Lipids are degraded by lipases to glycerol and fatty acids. Glycerol enters glycolysis. Fatty acids are degraded by beta oxidation, this process here. Now, I want you to look at the product of beta oxidation. Product of beta oxidation is acetyl coenzyme A. Acetyl coenzyme A will enter the Krebs cycle. So lipids are fed into glycolysis and the Krebs cycle. So you break them down to glycerol and fatty acids. Glycerol enters glycolysis. Fatty acids are broken down by beta oxidation to acetyl coenzyme. They're broken down 2 carbons at a time. And then those enter the Krebs cycle. What about proteins? Proteins are degraded by proteases. You then have amino acids. Amino acids have their amino groups removed by deaminases. These deaminases require the vitamin pyridoxal phosphate, otherwise known as vitamin B6. Vitamin B6 is an essential cofactor for these deaminases. Bacteria take the ammonium and just excrete it. In our bodies, what do we do with ammonium? Ammonium is very toxic to our bodies. What does your body do with ammonia? What does your liver do with ammonia? What's the main nitrogenous waste in pee? I don't speak mumble.
Ammonia?
No, urea. Urea. Your liver converts it to urea through the urea cycle. The urea cycle that is done in your liver. So we excrete urea because urea is much less toxic than ammonia. And problems with the urea cycle cause ammonium levels decline, and ammonium levels are very poisonous, particularly to the brain, because they interfere with neurotransmitter. The beta amino acids are then either shunted into glycolysis or the Krebs cycle. So all metabolic roads lead to Rome. They lead to glycolysis or the Krebs cycle, and eventually to the electron transport chain. That is catabolism. So what's the maximum yield of ATP per molecule of glucose for each of the following types of respiration? For aerobic respiration, we figured that out, it's 32. For fermentation, the maximum is going to be 3 or 4. That's about it. For anaerobic respiration, it's going to be more in the 24, 25 range. All right, now let's talk very quickly about anabolism. We've talked a lot about catabolism. You make ATP, now you use ATP to make things. Anabolism involves taking small molecules, linking them together to make bigger molecules. The precursors for those are taken from the Krebs cycle. They're taken from glycolysis, and they're taken from pyruvate and things like that. So some of these pathways are so-called amphibolic. Amphibolic. They make precursors that are also used for biosynthesis. And in fact, there are reactions in the cell that replace metabolites that you remove. These are called anaplerotic reactions. Anaplerotic reactions. Anaplerotic reactions are replacement reactions that replace metabolites that you remove from these pathways that keeps the pathways going. So acetyl coenzyme A is also used to make fatty acids, which are used to make lipids. To make carbohydrates, you take glycolysis and you run it backwards with 3 bypass reactions. There are 3 reactions in glycolysis that are not reversible. All the other reactions are reversible. That process is called gluconeogenesis. Very energy intensive process. Proteins, you withdraw precursors from both glycolysis and the Krebs cycle, and then you use ammonium, and you use molecules called transaminases to put the amino groups in. Those transaminases also use vitamin B6 as their cofactor, pyridoxal phosphate. And then nucleotides. Nucleotides are made from amino acids, and they're also made from precursors that come from another pathway that we haven't yet talked about. Oh, I've got 1 minute yet. Maybe I'll get a chance to mention it. Okay, said that, so on and so forth. Oh, here it is. Page 2. At the bottom, this pathway here, where you take glucose and convert it to ribose, this is called the pentose phosphate pathway. The pentose phosphate pathway. Pentose phosphate pathway will take glucose and use it to make ribose biphosphate. Ribose biphosphate is essential for nucleotides. That's how you make them. You make them from glucose and you make ribose biphosphate. Ribose biphosphate is essentially your building block for nucleotides. And then glucose is used to make cell walls, and some bacteria can make all the amino acids, like E. coli. Others need them in their diets, and blah, blah, blah. The ability of the cell to integrate molecule using molecule binding pathways to improve cell efficiency is known as molecule using and molecule building pathways. You take pathways that are useful for metabolism, you also co-opt them for anabolism. Okay. We'll start chapter 8 on Monday. Don't forget, next week we don't have lecture on Friday because we have the fall break. So Monday we'll start chapter 8, and chapter 8 is very complicated. Okay, Bree, that's all for today. Hope you're feeling well. You feeling all right? Bree, did you hear me?
Yeah, sorry.
You feeling all right?
Yeah.
Good. Okay. Then hopefully we'll see you soon.
Okay. Thank you.
All right, that's all for today. Have a good day. Be well.